Thank you, Michael. It doesn't say there, but I'm from the Department of Biochemistry at the University of Otago, and we're just trying to be useful. I'll just give you a sort of a flavour of the kind of work that we're doing. This is just a historical walkthrough. It's been used talking to other groups. I'm sorry, this is the wrong one. Okay, let's stuff that. We were chopping and cutting just before this at the back. We've obviously chopped and cut in the wrong way. How embarrassing. What have we been doing? And the answer is very much from the start we focused on the possibility of using antibiotics as tools to control the pathogen. And you might say, well, why antibiotics? And the answer is because other people use them to control similar pathogens in other jurisdictions. For example, in America, they use streptomycin and oxytetracycline routinely to control bacterial pathogens, including bacterial cankers on apples, pears, peaches, various other things. So do the Japanese. Sorry. So the reason why we decided to take an interest in particular in the antibiotics was because they are a proven technology which has got a history of effective application in similar contexts. There are lots of other possibilities that might work in the middle term, but these ones may work soon. They may be, to overuse a phrase, a silver bullet. So what are we going to do with the antibiotics? And what we wanted to understand was how the bacterium might respond to the antibiotic, whether resistance would be likely to emerge or not. In order to do that, we had to understand exactly what bacterium we were playing with. And one of the first things that we tried to understand was the status of the so-called PSA-V and PSA-LV. What was the relationship between these things? And these are the PSA-LV strains grown on a medium in which they become strongly fluorescent. They produce a yellow fluorescent protein. These are PSA-V. They're much less fluorescent. And this is the Japanese strain, and it's non-fluorescent. So it became obvious to us very early on that there were several different kinds of bacteria around. How can you sort that out better than just growing them on a plate? And one of the things you can do is sequence some of the genetic makeup of the organism. And we specifically sequence these genes here, which are routinely used by other people when they're comparing bacteria. You end up with this gorgeous looking slide. These are all different strains of bacteria. And for example, the thing that is sometimes called PSA-LV or PSD, I would prefer, is this group of strains here. And this is the sequence of one of the genes, the GLTA gene. And you can see the PSDs all have a blue nucleotide there. They differ in their sequence from everything else. And they have a blue one there, and they have a yellow one there. In multiple places, they are clearly a different bacterium. So there are two different sorts of bacteria clearly here. If you look at all of those genes I described, you can use computer programs to build a sort of a family tree of the organism. And these are the things we would like to call PSD strains. And these are the ones which are causing all the trouble, the PSA-V. There is Pseudomonas syringae thii. It's a pathovar from the tea plant. And there's Pseudomonas syringae mosprunorum, a pathovar from the cherry. And it's clear, therefore, that these are not just slightly different, they're miles different. This is one from the hazel. So, as far as we are concerned, one of the first things that we were able to do was set aside all of those PSD sequences and say, well, these are of no significance to us. They're of no interest to us. I'm sorry, this is the uncut down version. That's just looking at 6 of the genes. You know, maybe that's not an accurate impression of what's happening. This is what we've managed to do in the last month or so. It's the whole genome of the organism. This is the tomato genome along here. And these are the sequences of a PSA from Puke, 2 of them Puke, and a PSD strain. Each one of those tiny little blue flecks represents a DNA sequence. So that what this represents is a massive sequencing analysis to try and see how these things resemble each other, how they differ from each other, what makes them tick. And just to remind you, what we're trying to do is better understand how this organism is going to respond if we attack it with antibiotics. What's there of any interest to us? And the answer is, it's very obvious again that the PSDs are different. See that big white track there represents genetic material which is present in the tomato, present in the PSA-V. It's missing in the PSD completely. Here there's a sequence where the 2 PSA-Vs are very similar. They have that white track. The PSD is completely missing it. Very different beasties. And these things are very, very similar. They're not, in fact, absolutely identical, but we don't have time to go there. This now is 8 strains, and we have completed the genome of 8 strains, 2 PSDs, a Japanese PSA-V, and 5 assorted PSA-Vs, some from Italy, some from New Zealand. That represents 6.5 million base pairs, and there are 8 strains there. In total, that was about 30 gigabases of nucleotide sequence. It's an absolutely huge amount of information, and one of the problems with it is actually processing it. You know, it's one thing to complete the sequence, and it's another to make any sense of it at all. Galloping through, we have completed the sequence, and we've completed the assembly of that sequence, so we now have the complete genome of 3 critical strains, a Japanese, an Italian, and a PSD, and we can ask it questions about how might this organism respond to the antibiotics. And the way that you do that is to now question that sequence and say, you know, what have you got? And here you're looking to see, this is Pseudomonas syringae actinidae strep A gene from Japanese isolate. Is there anything like it in the New Zealand PSA-V? And the answer is no, that gene isn't there. And if that gene isn't there, then it can't be a problem to us. And secondly, this is now a different streptomycin resistance gene, and that one again, no hit at all. And so you can go through and see what has it got by way of the ability to respond to your antibiotic and what hasn't it got. The good news is that it's completely missing the strep A and strep B genes that might otherwise be a nuisance. And it's also completely missing the TET-A gene that might otherwise be a nuisance. And it's completely missing a little structure called a plasmid, which is a method that bacteria use to swap stuff with each other. And if it had a plasmid, we would be very worried, because it means that it's capable of talking with other strains and acquiring resistance. The PSA-V in New Zealand doesn't have any plasmids at all. So this, I mean, I realize there's not a lot of good news around. This is basically good news. It means that this organism, which is very sensitive to streptomycin and very sensitive to oxytetracycline, has not got a very good range of ways of becoming resistant to us. These are the people involved in doing it. There's me, there's Margie, there's this grouping called NZGL, and this is New Zealand Genomics Limited. they did the sequencing and they did the assembly, and we're just completing the annotation now. You might say, well, that's sort of organisms that have been sequenced and assembled in New Zealand. And people may choose to argue with that, but I'm happy to have that argument. These are the first organisms that have been sequenced and assembled in New Zealand. And they were sequenced and assembled very quickly because we recognize the critical nature of the problems that you face. We would like to be useful, and we realize that being useful means also being quick. So this was not only sequenced in New Zealand, it was sequenced damn fast in New Zealand. Where to from now? And the answer is, we have now that we have the complete genome, we have a method by which we can study resistance if resistance emerges. And that's something that I would be passionate about. If we're going to use streptomycin, if we're going to use oxytetracycline, we have to monitor to make sure that resistance hasn't popped up. Because if it pops up, we may be able to snuff it out. But if we just use the antibiotics and hope that it doesn't come off the trolley, then resistance could emerge, and after a few years, the antibiotics will become useless. So how can we use these sequences? And the answer is, now if we had a strain which inexplicably had become resistant, we can very quickly sequence it and define why it's become resistant. We can produce a PCR test that will be able to detect that resistance gene and tell us how widespread it is and be able to respond effectively. So they are useful tools enabling us to manage the use of the antibiotics going forward. What's the chance of this working? And the answer is, on the peach in North America, tetracycline has been used for 20 years, and as yet resistance has not emerged. And they simply find it enables them to manage bacteria, the particular bacteria that's troubling them. And certainly in the Western states, streptomycin and copper have both been used for 20 to 30 years, and they're still proving effective. So that I think there are reasons for hoping that if we approach this energetically, we may be able to manage it through to the middle term. I mean, given time, I hope that we can get plants that are naturally resistant, and that is obviously the better solution, but that will take time, and time we don't have at present. I'm sorry if that was a shambles, but somehow we managed to load Mark 1 instead of Mark 2.